Mr. Chairman, dear Tony, dear Philip, dear Günther, congratulations, by the way. Thank you for this honorific invitation, really. And you see already that when I am speaking, I am moving my hands, I am moving my lips, I am doing small movements with my body in order to talk to you. These are automatic movements. I am not thinking that I'm doing these movements, but nevertheless, I am doing these movements automatically. In fact, I do them subconsciously. What do I mean by subconsciousness? I don't think that I think, but I think. And this is done automatically. This is what I call subconsciousness. I don't think that I do, I don't think that I feel, but I do, I feel in an automatic manner. I hope you agree with this definition. If you don't, no problem. Subconsciousness then is different from what I will call pre-consciousness. Pre-consciousness is more or less the same, except that it's non-automatic. I don't think that I think, but I think in a non-automatic manner. I don't think that I do, I don't think that I feel, but I do or I feel in a non-automatic manner. And pre-consciousness and subconsciousness are certainly very different from consciousness. You know the definition of consciousness, very complicated. The best one probably is that of Sigmund Freud, who said, I'm conscious that I am conscious. Anyway, the way I see it, I define consciousness as follows. I think that I think. I think that I do. I think that I feel. I think that I am. Very complicated, but different from some consciousness. If one wants to approach the problem of the neural basis of consciousness, it's very, very complicated. But if we concentrate on subconsciousness then, perhaps we will have an answer. This is what I would like to do just now. And I have one candidate. What is the candidate? The basal ganglia, of course. So I will try to give you some information about the role of the basal ganglia in subconsciousness. But you will ask me the question, well, what do you mean by basal ganglia? Well, we all know the basal ganglia. The basal ganglia, if I may oversimplify, are characterized by three main features. First, segregation. You see here the cerebral cortex, the different territories of the cerebral cortex, the motor area, the associative area in purple, the limbic territory here in yellow. And these three territories are projecting to the basal ganglia here. Where is it? Well, here. The striatum, the globus pallidus, the subthalamic nucleus, at the upper part, at the middle part, and at the lower part of the basal ganglia. First characteristic, segregation. Then second feature, loop. Indeed, these three territories are projecting back to the cerebral cortex. I don't know how to use this. In particular at the level of the prefrontal cortex. So that there is a cortico-subcortico-cortical loop, right? Implying that the basal ganglia are closely linked to the cerebral cortex, of course. And finally, convergence. I missed part of the slide. It's not important. You know what's the volume of a cerebral cortex? Almost half a kilo. And the can you help? Can I have the sound then? Okay. Well, you have something like 20 billion neurons in the cerebral cortex, and you have only 200,000 in the pallidum and in the subthalamic nucleus. There is therefore a massive convergence. Thank you very much. Very skillful. So something I would be able to do. 20 billion neurons in the cerebral cortex. You see this massive conversion in the subthalamic nucleus, which is only 0.15 cube centimeter, which means that all the information which is located, which is contained in the cerebral cortex, more or less, are found in this little structure, the subthalamic nucleus. So what's the role of the basal ganglia? You know perfectly well the role of the basal ganglia, characterized by segregation, loop, and convergence. The motor role of the basal ganglia is well known. Three main features, to be brief, because I'm sure you know this by heart. First, execution. The intention of movement, the preparation of the movement, the initiation of the movement takes place in the cerebral cortex, first in the parietal cortex, then in the premotor cortex. And the basal ganglia have a very specific role, which is execution of movement, the automatic execution of movement of a motor program which can be explained here. This is, you see, my writing, something which is overlearned years and years of writing, in particular if you're a medical doctor. And you see here, this is my normal writing here. And at the upper part of this figure, I am writing only with 2 digits, so the letters are smaller. At the lower part of this figure, here, I'm writing with my arm. But interestingly enough, as noticed by several authors in the past, the pattern of my writing is more or less the same, right? This is what we will call a motor program, which is underlined by complex network that I won't describe here, probably somewhere in the cerebral cortex. First feature, execution. Second feature, selection. All the information in the cerebral cortex goes to the striatum, the role of which is to eliminate inappropriate movement in order to select the desired motor pattern, right? This is very well known. This is done in particular through collateral inhibition at this level. And the third feature is the most important, learning. I will give you 2 examples of activation changes in the striatum during motor learning. One experimental and the other in humans. Experimentally, I take the example of a study by, this is several studies, but I take this example by Anne Graybill and her collaborators. You take a rat, you place electrodes in the sensory motor part of the striatum. Then you place your rat here, in front of a T-maze, and the reward is placed at one of the extremities of the T-maze, here. At start of the study, the animal doesn't know whether the reward is placed on the right or on the left, but after several attempts, he learns how to, using trials and error, he learns that the pellet, the reward, is here. If you look now at the if you look now at the recording within the striatum, you see that at the beginning of the experiment, neuronal activity in the striatum is random. But after several attempts, when the animal has learned where is the reward, so he knows the trajectory, he has memorized, this is memorized, quoted memorized within the striatum, then the pattern of neuronal activity is really stereotyped with a peak of activity at start of the trajectory and a peak of activity when he gets the reward. The meaning of this is difficult, but it indicates that the striatum in these animals has the property to learn something. I give you another example taken from a study by Stéphane de Récy in Paris and collaborators in normal subjects. what these doctors, Loerici, did was to ask healthy subjects to learn a complex sequence of movement with the digits, something very complex that at start you don't know, but after a few attempts you learn how to do it. And what do you see? First, you see that, of course, at start the cerebral cortex is very much activated. But in the striatum, you see, here, you see here that what is activated in the striatum is first the associative part of the striatum, not the sensory motor part, which is which is here. But after several attempts, you see that the activation in the associative striatum is disappearing, almost disappearing, whereas it is increased in the sensory motor part of the striatum. In other words, in these healthy subjects, learning a new sequence of movement, then the sensory motor part of the striatum has in some way learned how to do this complex sequence of movement. We said execution of movement, we said selection of movement, shaping, gating of movement, in addition to the cerebellum, of course, and then finally learning. This is very well known as far as the motor role of the basal ganglia is concerned. But now what I would like to do is to show you that in addition to a role in motor activity, the basal ganglia have also an important role in cognition and in emotions. First, a study that has been done in our lab in Paris. You take a monkey. The anatomy of the basal ganglia is very well known. And for instance, here in the globus pallidus external part, some investigators in our group have made a detailed anatomy of this nucleus, and which can be distinguished into three different territories: the sensory motor territory, Jesus Christ, sorry, an associative territory, and the limbic territory, right? They are intermingled, but you can recognize these three different territories quite well in this structure. Then the idea was to see what happens if you manipulate this territory. This is what has been done by injecting a GABA antagonist in the globus pallidus in order to block the output system of the globus pallidus, right? What happens if you inject bicuculin in this case in the sensory motor territory? Well, you get dyskinesia, correct movement, or eventually of the dystonic type in the monkey. I won't show you the films. If you inject in the associative territory, you know what you obtain? Very interesting. You obtain something you see in children with ADHD. The animal is hyperactive and inattentive. Hyperactivity, inattention. Very, very interesting. And if you inject in the limbic territory, you observe stereotypies. What I mean by stereotypies, I am asking you the question, and I show you a film. I hope it will work. You see here this monkey, which is licking his tail, which is a normal behavior in monkeys, absolutely normal. He's washing his tails, in fact. But instead of lasting a few seconds, this is lasting for 1 hour. That is the time of the duration of the action of bicuculin, which has been injected in the limbic territories of the striatum, right? Of the globus pallidus, sorry. You see? This indicates that the manipulation of the limbic territories of these animals induce very repetitive behaviors, the meaning of which is difficult to understand. It's not really like tics, because it's continuous. It's not like compulsion. You see the eyes of the animal here. Here. He's not anxious at all. In fact, if you interrupt him by asking the animal to make something, to make a task, for instance, he does it perfectly well. He's perfectly conscious and very intelligent, not anxious. But I think that in these animals, too bad they don't talk, I think that in these animals have the urge to move. They cannot help moving. It's an automatic behavior which is subconscious. I give you trivial examples in human. You obviously know that patients with lesions in the substance nigra, in the striatum, in the pallidum, in the subthalamic nucleus, induce abnormal involuntary movement. This is what we see every day. But in addition, we see a lot of cognitive symptoms, let's say, because I don't want to enter into the details, frontal lobe-like symptomatology, and emotional disturbances. We just heard some of these emotional disturbances a few minutes ago. Perseverations, disinhibition, arithmomania, etc., etc. We are used to this. And if I concentrate on the problem of emotional disturbances, you may ask, well, these are retrospective studies, patients with lesions. Second, it's not really very specific. God has not placed the stroke or the tumors just in order to please the investigator. So it's not really specific. How can you really show that the basal ganglia is playing a real important role in the occurrence and the control of emotions? This is possible in two ways. First, you can provoke a given emotional state in someone and to see whether brain structures are activated in the brain using functional MRI, for instance. The other possibility is to manipulate the limbic territories within the striatum and to see whether you're able to reproduce these emotions, right? Two. You need the two. I don't want to stress NMR studies because there are so many which are shown in this Congress in particular. But I show you two studies, one, the upper part of this figure, by Dr. Zeki, who has studied romantic love. He took lovers and see what happens in the brain of these patients. And several studies of that type, for instance, one concerning beauty and attractiveness. And what is illuminated in the brain of these normal subjects? Of course, one, the limbic systems. But two, interestingly enough, a little bit the orbitofrontal cortex, the amygdala, etc. That is telencephalic limbic structures, but mostly the limbic territories of the basal ganglia. Isn't it unbelievable that here you see a strong activation of the ventral striatum, a structure which is small in the center of the brain, which is phylogenetically very old, which is predominant in the cat, in the snake, in the bee, which has almost no cerebral cortex. But have you seen Pigeon in Love? I'm sure you have seen. It's beautiful. So this simply indicates, using this MRI study, that if you get an emotion, automatic emotion, if I could say so, you are in fact activating the limbic territories of the basal ganglia. But this doesn't prove, of course, that the center of love, of beauty, of emotions is located in the ventral striatum. It simply means that these limbic structures within the basal ganglia are contributing. They are part of a network which is activating during such emotions. In order to really prove that the ventral striatum, for instance, is really playing a role, then you have to manipulate this structure and to see whether you are able to reproduce these emotions, love, for instance. And this is possible. This is possible using deep brain stimulation, because using deep brain stimulation, which is a reversible technique and adaptable technique, you are able to activate or inhibit a very restricted amount of brain tissue. For instance, in this case, you see these leads with several electrodes at the extremity, and you can target the subthalamic nucleus either posteriorly within the sensory motor part of the structure, or anteriorly, that is in the limbic part of the subthalamic nucleus. A lot of possibility. And this is why in some of our patients we see manic episodes, or such as this case from Grenoble, laughter. Does it work? No. You have seen this picture before. I think it's absolutely excellent. This patient has just been operated, and he's maniac, right? He has a laughter. It's more than mania. Anyway, so we see this quite often, but sometimes we can see horrible things. For instance, depression in these patients that we saw a long time ago with Dr. Bejani. This patient on the left is a Parkinson patient. She had been operated with two leads, two electrodes in the subthalamic nucleus. She's quite well. She has been nicely improved. But in order to improve her even better, even more, then Dr. Bejani tried to check the different contacts at the extremity of the leads. And when activating one of these contacts, suddenly, the second figures, the patients started to become perplexed, and then started a real acute transient depression. She said, I want to die, I'm fed up with life, and you don't need to be a doctor to understand that this patient is depressed, the third picture. And then as soon as the current was interrupted, there was a slight rebound phenomenon, so that the patient was hypomaniac for a few seconds. And interestingly enough, the electrode was located in the substantia nigra pars reticulata, and we know that this was located in the limbic part of this output system of the basal ganglia. Why? Simply because this patient accepted to have a PET scanner, and using the PET scanner, the stimulation of that site, precisely, which was not the therapeutical site, was able to induce, to activate all the limbic territories within the brain, in the thalamus, in the amygdala, in the orbitofrontal cortex. So the message here is that using this technique, the manipulation of the limbic relays within the basal ganglia can induce strange emotion of the mania type or the depression type. But you can see also rage, many things like this. This technique has been used to treat patients. This is perhaps something which is new for the MDS psychiatry. For instance, several results have been obtained, good results have been obtained here and there in Gilles de la Tourette's disease patients by stimulating various limbic relays in the basal ganglia. In this first experiment, this was 9 years ago, we did the first patients, and you see the electrodes were placed in the limbic territories of either the internal pallidum, which is the limbic territory of the pallidum, or in the parafascicular nucleus, which is a limbic relay within the thalamus. And the results are unbelievable, really unbelievable. All the patients who have been operated 8 years ago can be considered to be normal at that time. I would be delighted to have the experience of other investigators. For instance, this is typically a patient with Gilles de la Tourette's disease, you see. Okay. You all know these patients. We see a lot in Paris where we have a reference center. And during the sham, after following pallidal stimulation, everything is disappeared. The same for OCD patient. Obsessive compulsive disorders, for reasons I cannot describe, because we have several experimental data in monkeys. You can use stimulation of also limbic relays in the basal ganglia. There are several studies going on. We did some in a multi-center study in France with some, promising results. Not in all patients, but in some patients. I know 2 patients who are totally relieved, really, because these are extremely severe cases, resistant to any kind of therapy. I show you these extraordinary patients from Dr. Paul Crac in Grenoble. This guy has an obsession of snake, diseases, deaths, etc. And Dr. Crac is asking him to repeat the test sentence. The snake is dead, it is buried in the cemetery, it had a heart attack. I hope it will work.
Est-ce que vous pouvez me répéter la fameuse phrase?
Non, impossible.
Le serpent, il est décédé,
Non.
elle est enterrée au cimetière.
Non. Je peux pas du tout. Autant je pourrais...
Même si c'est moi qui l'ai dit, ça vous stresse?
Non, ça me stresse. Je veux dire le serpent. Je peux dire déjà le serpent.
After 2 minutes
Pas
stimulation,
grand-chose, il est mort, il a une crise cardiaque, non. A priori,
and he can repeat this sentence normally, right? This is one of the good cases, of course. So now it is time for me to conclude. Can I get the slides off, please? These are the collaborators who participate in most of the study. Can I have the light? It is time to conclude. We're dealing with the basal ganglia, small structures which are enormous in the pigeon. I like this one because the cerebral cortex in the pigeon, called the pallium, is very small. And these are structures which are phylogenetically very old, very, very old. In the bee, there is the most cerebral cortex. And you have seen how important they are. And I would like to leave you with 2 messages. The first message, the basal ganglia are not only involved in motor behavior, but also in cognitive and emotional behavior. So that the famous statement of the late David Marsden, our friend David, the role of the basal ganglia is the automatic execution of learned motor plans, should be perhaps slightly modified as follows. The basal ganglia are involved in the automatic execution of learned motor, cognitive, and emotional plans. That's the first message. The second message is more related to subconsciousness. Why have I added the word subconsciousness? I don't know. But if you remember the definition I gave of the word subconsciousness, I don't think that I think, I don't think that I do, but I think I do automatically, then you will have to accept that the basal ganglia is really playing a role in subconsciousness. But the fact, the basal ganglia, in fact, are not totally isolated. Remember, they are closely linked to the cerebral cortex. There is a subcortico-cortical loop that we described briefly. Therefore, there is a permanent dialogue between the cerebral cortex and the basal ganglia, so that the basal ganglia are predominantly involved in subconscious automatic behaviors, and the cerebral cortex predominantly involved in preconscious non-automatic behaviors. And in fact, you have here, in this audience, an exquisite demonstration of what I wanted to say about subconsciousness and consciousness. Just by attending this audience, just by looking at this lecture in the honor of David Marsden, in fact, all what I said was said subconsciously. You see, I'm so used to teach. In French, usually, it's much better. It is done subconsciously. It's like playing tennis. It's like driving my car. This represents 98% of my life. And yours. For some people, it may be 97. For others, it might be 100%. So, for a while, I would like to leave my subconscious strategy now and to turn my attention to a preconscious strategy. And I will leave all my routines, I will leave all my habits, I will concentrate on myself, and I would like to say, long life to the movement disorder society.